Hello, and welcome to the Rejuvenation Roundup podcast, where we explore the latest in life extension and anti-aging science with a dive into a month's worth of insights and new breakthroughs. This podcast is a combined effort of the Life Extension Advocacy Foundation, which operates Lifespan.io, and Future Grind, a podcast that explores the ethics and impact of emerging science and technology. I'm Ryan O'Shea, and I'll be your host. We are nearing the end of 2019, but before we get there, let's look back on what happened in the month of November. New episodes of the Life Extend show have been released on our YouTube channel, and they feature a new member of the X10 crew. One episode focuses on the depiction of life extension technology in fiction, and another recaps our 2019 conference. Here's a sample of that.
Last July, Life Extension Advocacy Foundation had its second Ending Age-Related Diseases Conference, or EARD for short. It was a great event focused on the latest exciting developments in the field of rejuvenation biotechnology, packed with awesome speakers and talks. If you've been following this field, you might know that in the past 10 years or so, scientists studying aging have completely changed their minds about it.
There's much more interest in aging, I think, these days on a research level and on a practical level and a medical level, in part because the field has really developed tremendous breakthroughs. We understand things now we never understood before. We've been able to accomplish things we've never been able to accomplish before.
There has been a shift in paradigm, a shift in our concepts of aging as a plastic process.
When I was in medical school 6 years ago, I distinctly remember sitting in a lecture hall on geriatric medicine where the lecturer explicitly said, but of course no one is trying to develop drugs for aging, indicating that it's something that we really couldn't do anything about. And yet this very year, that same medical school broke ground on a longevity institute on the campus.
Everybody used to think that aging was inevitable, that rejuvenation was the stuff of myths and sci-fi. But that notion has been completely turned on its head.
And particularly thanks to the breakthroughs in our manipulation of aging in model systems, how we can now extend lifespan, delay aging, retard age-related processes in animal models, including mammalian models like mice. There's been a great awareness, or a much greater awareness now, that we can manipulate aging in animals, and therefore, why not in humans?
Very many people sort of changing their ideas about aging as being something intractable and hopeless and forlorn into something exciting, that we can actually do something about.
We've seen a complete 180-degree reversal in public and academic sentiment around this. We've really come to understand what it is that causes us to age, and seen some evidence of actually attacking those things, and seen extension of lifespan of mice, healthy lifespan, it's important to note.
Although there is disagreement in the field about how soon rejuvenation will become reality or about the effectiveness of the treatments currently proposed or under development, consensus seems to be that a future without aging is a matter of when, not if.
We will ultimately achieve full medical control over aging. I would predict that would happen this century sometime.
Translating some of the findings from animal models to humans, I'm optimistic about it. I'm optimistic about longevity drugs coming into market in humans. So I do think that's going to happen in the foreseeable future. Now, complete control of aging, that's something we cannot achieve yet in animal models. So to think that we're going to do it in humans, well, I don't think there's any reason to think we cannot do it. I think in theory it's possible. There's animals that don't age at all. So in theory it is possible, but from a practical perspective, how we're going to about do it, we have no idea really. So I don't think it's going to happen in the foreseeable future. I think it will happen one day. Whether that's going to be 50, 100, 1,000 years from now, I don't know, but I do think it will happen one day.
I don't think that there's any necessary limit, at least not from a physics or biological perspective. Medically, things take a very long time in order to test properly. So I think that we will know a lot more about the degree to which we can prevent diseases of aging after we start really establishing a paradigm where we can test clinically drugs for prevention of disease. I think it's going to still take a little while before that's done routinely.
Researchers and advocates of rejuvenation also agree on the fact that focusing on the prevention and the treatment of aging is extremely important for humanity as a whole.
Aging is directly or indirectly the greatest cause of suffering, disease and death in the world.
There is quite a lot of consensus that these diseases have a common cause, and this common cause is aging.
So we really need to tackle aging to address the graying of the population and to eradicate or at least ameliorate the various diseases and degenerative conditions that are associated with aging.
Personally, I would like to think that, any person over time would gravitate to the idea that life is good and is worth living, and thereby, as a natural extension of it, I guess pun intended, that more life is a good thing, not just for you, but for the people you love and anyone who's wanting to be alive.
Oh, it's been wonderful. I'm very, very impressed with everything, particularly all the excitement. A lot of people, a lot of interest in the field. It's really a big change from even a few years ago. There's a lot of positive vibrations in the field of aging. A lot of people wanting to really go for it. So it's been a wonderful conference, and it's been a wonderful impression.
The LEAF team has done a fantastic job. I love the grassroots efforts and advocacy. We even co-hosted a pre-conference workshop that I gave on how to build a pharmaceutical company in the aging space, and we donated all the proceeds from that to LEAF as an indication of the quality work that I think they do. So very excited about the conference, very happy that it's a 2-day event, love the panel of speakers, and it's a great opportunity to get a crash course on everything exciting in the space.
Yeah, this is a great conference, enthusiastic, smart people, brilliant people, a lot of cutting edge things here, including things I really hadn't heard about before. I think the future of aging is safe with this group. I think we're doing a great job at this gathering.
Just like EARTH 2018, the conference this year brought together some of the most famous researchers of the field working to understand aging and to bring it under full medical control. And they discussed some of the hottest topics in the field, like epigenetic clocks or senolytics, that's a big favorite, thymic regeneration, and a lot more. But you should definitely go to our YouTube channel and check the full talks there.
Speaking of EARD, we've now released the final video from our conference, featuring João Pedro Magalhães of the University of Liverpool, discussing potential failure points in longevity research and how to overcome them. Here's a bit of that.
And thank you to the organizers for the invitation and for organizing this conference. I mean, one of the things that actually you'll understand relates to my talk is how impressive it is to see a conference like this one. When I started in the field 20 years ago, I don't think we would be able to have an ending age-related diseases conference with so many people, and really the excitement and all of the activity, both academia, industry, even at government level now in aging is very exciting. Well, I guess for a conference on aging and ending age-related diseases, I don't need to remind you of the importance of tackling aging. I guess the point that we're all convinced of is that we need to tackle aging. We need to develop interventions that allows us to at least retard or delay the process of aging, that allows us to reduce or even eliminate age-related diseases, which are now the major burdens of medicine and of society. And if we could do that, if we could develop one way, as I think in the previous discussion, was if we could at least develop one way of retarding aging, now that would have huge medical, health, social, and of course financial benefits for whoever was able to do that. So we now know that the process of aging is plastic, and that means that we can manipulate aging in model systems, in animals, by genetics and by interventions. We can knock out individual genes, and this has considerable effects in the level of aging, both accelerated aging and retarding it. So I think this has really been a revolution in the field, and one of the reasons why there's so excitement about the field of aging now. So one of the things I actually started when I was a PhD student many years ago was a database called the GeneAge Database of Age-Related Genes. I'm sure many of you are familiar with it. But what I think is quite impressive is how much we have advanced in our understanding of the genetic basis of aging. So we now know of over 2,000 genes in model systems, so in mice, flies, worms, and yeast, that when individually manipulated have a significant impact on aging or longevity. So I think this is remarkable. So in worms alone, you can knock out a single gene, or it's an H1 allele to be more specific, and you increase lifespan by 10-fold. And in mice, you can knock out a single gene, I think one of the growth hormone receptors, and you can extend lifespan by up to 50%, as well as retard a variety of age-related diseases. And again, I think this is a remarkable breakthrough in the field. I'm convinced that some of the pioneers, like Cynthia Kenyon, Tom Johnson, will receive the Nobel Prize for these advances at the genetics of aging. And that's one of the reasons why really there's been all of this excitement about the field of aging that we see reflected here in this conference. Likewise, there's a lot of advances in terms of pharmacological interventions in aging. So another database we have is the Drug Age Database of age-related drugs. We've actually just updated 2 days ago, just before I came here. We now know of over 500 drugs or compounds that in mortal organisms significantly extend lifespan. Now, the effect sizes of drugs and compounds are not as great as those you see from genetic manipulations, but it's still very remarkable how much we've advanced in pharmacological manipulations of aging. And again, it's one of the reasons for all the excitement in the field. And so it's not surprising that we now want to harness all of these advances at the genetic and pharmacological level to develop interventions, to go into the clinic. And so we have this growing and exciting anti-aging biotech sector. So really the point is that there's been a lot of advances in model systems to understand how we can manipulate aging. And not surprisingly, there's a lot of academic labs and companies trying to capitalize on that. So trying to harness what we've discovered to develop human therapies, which again, if we could make that work, would be tremendous, which would be revolutionary, as been pointed out before. Now, what I wanted to focus, however, today is, but what if we fail? So the question I posed myself before I came here, and that I want to pose to you is, what are the possible reasons for us to fail? If 20 years from now, nothing of this have happened, we have actually not achieved in developing interventions based on what we know of the aging, what are the possible reasons for that? And I was thinking about that, and I came up with a few possible reasons. Maybe there's others you can think of, and if so, I'm happy to discuss afterwards. But my idea for this exercise is, okay, if we can think of reasons for why we will fail or why we may fail, then maybe there are ways then of mitigating these possible reasons for us to fail. So I'll give you a couple of reasons. Those I thought would be more obvious, or those I could think of. Again, maybe I'm missing something. And then I'll give some ideas as well as how we can mitigate risks based on these things that could go wrong. So one of the first reasons I think we could fail is, well, maybe what we know about aging is wrong. The other possible reason for failure would be that, well, we know a lot about aging in animal models, but maybe this does not reflect what happens in human beings. Maybe we human beings, maybe the hallmarks of aging are correct, maybe the reasons for the causes of aging we think are important in animal models, but perhaps they're not in humans. And there's reason to think that may be the case, actually. Some evolutionary biologists have pointed out that, well, we know a lot about manipulations of aging in short-lived animal models. But of course, maybe what makes a mouse live longer, or makes a short-lived animal live longer, well, that's not going to work in humans because we're already long-lived. The question then is, all right, so that's not very optimistic, is it? But how can we improve our odds? We do know that there's a lot of excitement about all of the genes and drugs that can modulate longevity in model organisms. I think that, this has been fascinating and it's been incredible. It's really generated a lot of buzz about the field. Having said that, I do think it's important that we try to anticipate failure, we try to predict why we could fail so we can then minimize risks. At the level of, okay, maybe we don't understand aging all that well, particularly human aging. As I mentioned before, humans are not just big mice or gigantic worms. There are differences between species. So what works in mice does not necessarily work in humans. I don't know, I think we've cured cancer in mice hundreds of times, but it doesn't necessarily mean we've cured cancer in humans. So what works in mice does not necessarily work in humans. And again, we have this very long experimental time in our field in particular of aging, which I think is a big bottleneck. I think is a big issue that is not easy to change, no matter how much technology or money we put into it. Having said that, there are ways, I hope I persuade you that employing data-driven methods and integrating human data of trying to minimize risk and trying to improve the odds that what we're discovering from model systems will work in human beings.
You can catch the full video, as well as other talks and episodes of the Life Extend show on our YouTube channel. Visit lifespan.io/roundup for more information. And now for our research roundup. In early 2019, a study in China looked at the microbiomes of healthy, long-lived people over the age of 90 and observed that such people had gut microbiomes that were similar to those of younger people. Another study showed that transplanting gut microbiomes from young to old mice reverses immune decline, and a recently published paper builds on this. This new paper explores modifying the gut microbiome to increase production of butyrate and brain-derived neurotropic factor in order to improve cognitive function. This study builds on an increasing amount of evidence that the gut microbiome has a significant influence on health and thus longevity. It also strongly suggests that adjusting the types and numbers of bacteria in the gut to more closely emulate a youthful microbiome may be a viable approach to improve health in older people. Recently, the first attempt in the United States to use the gene editing tool CRISPR to combat cancer appears to have gone well, according to the initial results of a University of Pennsylvania study to determine safety for the approach. This development is somewhat behind the times, however, given that Chinese scientists already tested this approach in cancer patients. It has taken over 2 years for the regulatory system in the United States to approve testing, which highlights the urgent need for reform in the approval system. Otherwise, the US could fall further behind in developing new medical innovations and therapies. That said, the arrival of CRISPR gene editing approaches in humans in the US is very welcome, as they have the potential to be an effective solution to cancer. Not only that, this trial may also open the door for CRISPR therapies against other diseases as well. A team of researchers, including renowned geneticist Dr. George Church, has published a new paper demonstrating the mitigation of multiple age-related diseases. Rejuvenate Bio published the data, showing how it used a gene therapy to selectively and simultaneously target 3 longevity-associated genes. We discussed this paper in greater detail in November's Journal Club, which you can find on our website. There's plenty more updates in longevity science that we don't have time to cover here, so to find out more about these or other advances, visit lifespan.io/roundup. And now for some news nuggets. An international team of scientists have issued a publication urging the World Health Organization, governments, and the medical science community to work together to develop classifications and staging systems using a new framework as a basis for diagnosing and treating age-related diseases. This is an important step in regulatory matters, and if successfully adopted by the WHO, could have a dramatic impact on the development of drugs and therapies aimed at effectively treating age-related diseases. Underdog Pharma, a spin-off company of the SENS Research Foundation, was launched with the lofty goal of eradicating atherosclerosis. Atherosclerosis is the leading cause of death worldwide and currently has no effective solution. Current medicine has 3 broad approaches: lifestyle changes such as diet and exercise, drugs that reduce the accumulation of cholesterol and thus slow down the rate of plaque formation, and surgical procedures such as bypass surgery, which attempt to solve the problem with brute force. None of these approaches offer an actual cure for the disease, which is why Underdog is developing a therapeutic approach that could potentially address the problem by removing a harmful lipid known as 7-ketocholesterol to halt progression of the disease and potentially even reverse it. In a study from researchers primarily based in Philadelphia, rapamycin, an FDA-approved drug that is used to combat organ rejection during donor implants, was shown to thicken skin and increase collagen when administered through the skin with DMSO. The researchers suggest that these initial results show that rapamycin has potential in addressing skin aging in humans and could be developed for therapeutic use. The LEAF Scientific Advisory Board has grown recently with the addition of Dr. Natasha Vita-Moore. Dr. Vita-Moore is an author, humanitarian, and innovator whose work focuses on longevity and enhancement. She was the lead scientific researcher on the Memory Project, which involved the long-term memory of C. elegans in cryonics. Currently, she is a professor of humanities in ethics, business, and entrepreneurship, and serves as the executive director of Humanity Plus, an international organization that advocates the ethical use of emerging technologies to enhance human capabilities. That's it for this episode of the Rejuvenation Roundup podcast. Thank you very much for spending another month with us and for your help in the fight against age-related diseases. Whether you're donating, spreading the word, or simply listening to our content, we appreciate your help. Remember to subscribe, leave a review, and post about us on social media. This will increase our reach and introduce more people to the importance of life extension science. Don't forget, you can get additional deep dives into science, technology, and futurism on the Future Grind podcast. Find out more at futuregrind.org. Once again, I'm your host, Ryan O'Shea, and on behalf of the team at Leaf, we wanted to thank you for joining us. We hope to see you next time on the Rejuvenation Roundup podcast.